In this chapter, so far we have studied that to increase the crop production, we must adopt good plant breeding techniques and animal husbandry management for fulfilling the demand of the population. But just going for animal and plant products is not enough. Along with plant and animal products, we can use the microbes as a source of nutrients. And this use of microbes as a source of nutrient is called as single cell protein, SCP. In SCP, we culture the microbes for their nutrients. A very good example of a very good source of protein, minerals and vitamins along with fat and carbohydrate is spirulina. It's a blue-green algae. And this spirulina can be used in the form of human food supplement as well as animal feed supplement. And to grow this spirulina, we need only the waste product like wastewater coming from potato processing plants or agriculture leftover like straw, molasses or even the sewage water. So by this process, we are cleaning the environment. It is called as bioremediation. Another very good example is Methylophilus methylotrophus. It is a bacteria which can produce 25 tons of proteins from the waste product in 24 hours. Along with that, we can use the microbes like agaricus, the mushroom, and other good microbes, bacteria for our food supplement. Now let's switch over to another field. A very new field is tissue culture. So tissue culture along with traditional method is used for developing new crop varieties. In tissue culture, we used a technique by which we can take a small part of a plant and culture it in the lab under aseptic condition in nutrients. There is a concept called as totipotency. It is the ability of every cell in the plant which can propagate to develop a new plant. For this, we require special nutrient media which contains sucrose, inorganic salts, various vitamins, proteins and most important growth regulators like auxins and cytokinins. So what we do? These are the steps for tissue culture techniques. We excise a small part of the plant, which can be any part. It may be a root, a shoot, or even a leaf. And this part, which is excised, is called as explant. This explant is placed in a nutrient medium under aseptic condition and it is allowed to grow mitotically into a mass called as callus. This callus is now transferred into another medium where it shows differentiation into small plantlets. These plantlets are then transferred into pots and later on, after hardening them, they are transferred to the field. So this tissue culture method is a very fast method to propagate the high quality plants. So what we do? We can take any small part and we can produce a large number of the clones from it. That's why it is called micro propagation. And whatever clones we are produced, they are called as soma clones. So tissue culture method is very commonly used to cultivate high quality tomatoes, bananas and even good quality apples. And another good thing about tissue culture is that we can even take a small part of the diseased plant, that is meristem part for tissue culturing. Even if it diseased, that particular part, apical meristem, is free from the disease. That's why we can, from a disease plant also, by tissue culture, you can cultivate disease-free plants. So this is a very good option. Now, apart from tissue culture, we can go for another method that is called as somatic hybridization, where two different species of plants can be propagated by culturing them. Only their protoplasm can be fused in the culture medium by using a chemical called as PEG, polyethylene glycol, which dissolves their cell wall and the protoplasm fuses, and that is a hybrid protoplasm. And the plant which is produced is a somatic hybrid, which has the properties of both the types of the plants. So this method of developing the somatic hybrids by tissue culture method is called as somatic hybridization. By somatic hybridization method, we can develop very good quality plants which have the property of two different plants. A very good example is the development of tomato, where the properties of tomato and potato are in the same plant. So with this, we can say that all these plant breeding methods, animal husbandry methods, and modern culturing methods are very important strategies for increasing the crop production. So with this, we complete this chapter, and this chapter is very important. Please go through it, and if you have any doubt, you can ask me. Thank you very much.